The process of cell division. Where do you find a copy machine in your body? The genetic information that is passed on from one generation of cells to the next is carried by chromosomes. Every cell must copy its genetic information before cell division begins. Each daughter cell gets its own copy of that genetic information, and cells of every organism have a specific number of chromosomes. Prokaryotic cells lack nuclei. Instead, their DNA molecules are found in the cytoplasm. Most prokaryotes contain a single circular DNA molecule or chromosome that contains most of the cell's genetic information. In eukaryotic cells, chromosomes are located in the nucleus and are made up of chromatin. Sister chromatids, here's a centromere that connects them, and these are duplicated chromosomes. They're made up of a supercoil, which is made up of nucleosomes with histine proteins that are all wrapped around the DNA double helix. Chromatin is composed of DNA and histone proteins. DNA coils around histone proteins to form nucleosomes. Nucleosome. The nucleosomes interact with one another to form coils and supercoils that make up chromosomes. The prokaryotic cell cycle is a regular pattern of growth, DNA replication, and cell division. Most prokaryote cells begin to replicate or copy their DNA once they have grown to a certain size. When DNA replication is complete, the cell divides through a process known as binary fission. Binary fission is a form of asexual reproduction during which two genetically identical cells are produced. For example, bacteria reproduce by binary fission. Cell membrane with the DNA. The DNA duplicates, and then the cell membrane indents, and the cell divides into two identical cells, which were also identical to the parent. The eukaryotic cell cycle consists of four phases: G1, S, G2, and M. Interphase is the time between cell divisions. It is the period of growth that consists of the G1, S, and G2 phase. The M phase is the period of the cell division. So G1 is cell growth, S, DNA replication, and G2, preparation for mitosis. And finally, for cell division, mitosis happens, and then cytokinesis in the M phase. In the G1 phase, cells increase in size and synthesize new proteins and organelles. So they're producing enough of the proteins and enough of the organelles to eventually split. In the S or synthesis phase, new DNA is synthesized when the chromosomes are replicated. This is the body's most efficient and effective copy machine. In the G2 phase, many of the organelles and molecules required for cell division are produced. In eukaryotes, cell division occurs in two stages: mitosis and cytokinesis. Mitosis is the division of cell nucleus, and cytokinesis is the division of the cytoplasm. Mitosis and then cytokinesis. What happens during the four phases of mitosis? This is an intro. During prophase, the DNA condenses and chromosomes become visible. Outside the nucleus, a spindle starts to form. During metaphase, the centromeres of the duplicated chromosomes line up and spindle fibers prepare to separate them. During anaphase, the chromosomes separate and move along spindle fibers to opposite ends of the cell. During telophase, the condensed chromosomes, which were distinct, begin to relax into a tangle of chromatin. During prophase, the first phase of mitosis, the duplicated chromosome condenses and becomes visible. And centrioles move to the opposite end and spindles forming. The nuclear envelope and chromosomes are duplicated and centromeres right here connecting the two. The centrioles move to opposite sides of the nucleus and help organize the spindle. So centrioles, and they help organize the spindles and begin to separate the two nuclei. The spindle forms and DNA strands attach at a point called their centromere, in the center, usually in the center. The nucleolus disappears and nuclear envelope breaks down. During metaphase, the second phase of mitosis, the centromeres of the duplicated chromosomes line up across the center of the cell. The spindle fibers connect the centromere of each chromosome to the two poles of the spindle. During anaphase, the third phase of mitosis, the centromeres are pulled apart and the chromatids separate to become individual chromosomes. Chromosomes separate into two groups near the poles of the spindle. During telophase, the fourth and final phase of mitosis, the chromosomes spread out into a tangle of chromatin. Nuclear envelope starts reforming. A nuclear envelope reforms around each of the clusters of chromosomes. The spindle breaks apart and a nucleolus becomes visible in each daughter nucleus. So a summary of the four phases of mitosis. The first phase of mitosis, the duplicate chromosomes condense and become visible. The second phase of mitosis, the centromeres of the duplicated chromosomes line up across the center of the cell. That's metaphase. Anaphase, the third phase of mitosis, the centromeres are pulled apart and the chromatids separate to become individual chromosomes. Phase, and the last phase, the fourth and final phase of mitosis, a nuclear envelope reforms around each cluster of chromosomes. So how do daughter cells split apart after mitosis? Cytokinesis is the division of the cytoplasm. Cytokinesis completes the process of cell division. It splits one cell into two. The process of cytokinesis is different in animal and plant cells. The cell membrane is drawn in until the cytoplasm is pinched into two equal parts. Each part contains its own nucleus and organelles. In plants, the cell membrane is not flexible enough to draw inward because of the rigid cell wall. Instead, a cell plate forms between the divided nuclei that develops into cell membranes, one for each side. A cell wall then forms in between the two new membranes. So in summary, the stages of cell cycle: interphase, prophase, metaphase, anaphase, telophase, and then finally cytokinesis. And that gives you two cells. From one becomes two. And then each of these grow and then also divide.